Imagine a patient, right? They're sitting in a brightly lit exam room, and on the surface, they feel, completely fine.
Right, maybe just a vague ache in their lower back.
Yeah, exactly. The kind of ache you just brush off as a pulled muscle from lifting something heavy. But deep inside their abdomen, hidden entirely from view, there is a ticking time bomb.
It really is a time bomb.
It's an abdominal aortic aneurysm, or AAA. And if you look at the mechanics of it, it's this progressive, totally silent ballooning of the body's main artery, the aorta. Every single time the heart beats, this massive surge of pressurized blood just pounds against a localized weak spot in that arterial wall.
And that wall just gets thinner and thinner.
Right. The balloon stretches further, and if it stretches too far and bursts, the mortality rate is just staggering. And the truly wild part of this scenario, right now in modern medicine, there is literally no pill to fix it.
None. Zero.
If you have an aneurysm, the standard of care is essentially, watch and wait until the bomb gets so big that a surgeon has to go in, open you up, and physically graft a synthetic tube into your blood vessel. It's an incredibly invasive, high-stakes gamble.
Yeah, it really highlights this glaring blind spot in vascular medicine. I mean, we spent decades understanding the physics of an aneurysm.
Yeah.
We know the extracellular matrix degrades and the vessel wall loses its structural integrity.
Sure.
But the underlying cellular chain reaction, like the exact sequence of molecular events that strips away the artery's defenses in the first place, has been this massive black box. Treating it surgically is kind of like showing up to a collapsed building and just, patching the concrete without ever asking why the steel beams dissolved to begin with.
Okay, let's unpack this, because today we are taking a deep dive into a 2026 paper from Nature Immunology that completely rewrites how we fix this time bomb.
It really does.
It's basically a brilliant biological detective story. We're going to trace the evidence from human single-cell sequencing all the way down to a brand new therapeutic peptide. And for you listening, don't worry, we are explaining every single mechanism along the way. No jargon left behind.
Absolutely. And the entry point into this detective story actually challenges a pretty long-held assumption about the immune system. Like, when we hear the word macrophage, the standard mental model is the body's circulating garbage truck.
Right. White blood cells born in the bone marrow that rush to an injury and chew up debris.
Exactly. And in the context of an aneurysm, these circulating macrophages have historically been viewed as the primary villains.
Because they secrete those destructive enzymes, right. The matrix metalloproteinases that literally digest the elastin holding the blood vessel together. They're the demolition crew.
That is the traditional view, yeah.
Well,
but the researchers in this study focused on a completely different lineage. They looked at vascular tissue-resident macrophages, or VRMs.
okay, VRMs.
Right. And these cells are not born in the adult bone marrow. Their origins actually trace all the way back to embryonic development, specifically the yolk sac and the fetal liver.
Oh, wow. So they've been there since before you were born.
Exactly. During fetal development, they migrate to the blood vessels and embed themselves permanently in the adventitia, which is that tough outermost layer of the artery.
I love the visual of this. So think of the adventitia as the outer boundary line of this vast national park. These VRMs are the dedicated, localized park rangers.
That's a great way to look at it.
Right. They aren't driving in from the city when there's an emergency. They literally live in the forest. And to distinguish themselves from those destructive circulating demolition crews, these embedded rangers wear a very specific molecular badge on their uniform.
The Live1 receptor.
Exactly. It's a surface receptor called Live1. So you have these Live1 positive rangers actively patrolling the borders, keeping the structural integrity of the blood vessel intact against that constant pounding of blood pressure.
And the clinical correlation regarding these specific rangers is what really kicked off the entire investigation.
What did they find?
Well, the research team took aortic tissue from human patients who had suffered these aneurysms, as well as tissue from mouse models, and they utilized single-cell RNA sequencing.
Which is where you look at individual cells to see what genes are turned on.
Exactly, cRNA-seq. When they mapped out the cellular landscape of a healthy aorta versus a diseased one, this stark anomaly jumped out. Among all the clusters of macrophages, the specific population of Live1 positive park rangers had almost completely vanished in the diseased tissue.
Just gone.
Basically, yeah.
Okay, but here is where I have to push back a bit, because the methodology gets tricky here. Just because you arrive at a crime scene and the security guards are missing, that doesn't mean their absence caused the break-in.
The old correlation versus causation trap.
Exactly. How do we know these rangers didn't just flee a sinking ship? The tissue in an aneurysm is highly inflamed and toxic. Maybe the local environment just killed them off as a byproduct.
That was the exact critical thinking hurdle the researchers had to clear. So to prove that losing these cells actively drives the disease, they had to isolate the variable. They moved to a specific genetic knockout model.
Okay, what kind of mice?
They used Angi-LD-LRKO mice. Basically, mice genetically prone to high cholesterol, mimicking human lipid profiles, and they infused them with angiotensin II to drastically spike blood pressure.
So they basically built a controlled environment mimicking a human with severe cardiovascular risk factors, a perfect storm.
Precisely. But here is the critical intervention. Within that high-risk environment, they selectively deleted only the live-1 positive tissue-resident macrophages. They left every other cell perfectly intact.
And if the Rangers were just innocent bystanders, deleting them shouldn't change anything?
Right, but the results were catastrophic. Without those specific macrophages, the mice had a drastic reduction in survival. Under the microscope, the aorta showed massive ballooning, and the elastin fibers were utterly decimated. Yeah, rupture rate skyrocketed. So this definitively proved the concept. The Rangers aren't just fleeing. Their physical presence is profoundly protective.
Okay. So losing the VRMs actively accelerates the disease, but I want to throw a question out to you listening. If you're this microscopic park ranger, what exactly are your tools? You're a single cell facing down systemic blood pressure. You can't just hold the vessel together with your bare hands. What are you using to protect the wall?
Well, to find that tool, they shifted from genomics to proteomics. They looked at the actual proteins deposited in the matrix, and they discovered these localized macrophages are prodigious manufacturers of a secreted protein ccalled Sparkle1.
Sparkle1 or SC1 for short, right?
Right. And when they measured SC1 protein levels in both human and mouse aneurysms, they found the levels had absolutely plummeted.
But wait, earlier they did RNA sequencing. Did the RNA instructions for SC1 disappear too?
What's fascinating is the discordance. The messenger RNA, the blueprint for SC1, was completely unchanged. The factories still had the blueprints, but the final protein was absent from the front lines.
So the stress of the aneurysm is somehow stopping the actual protein from being deployed. It's neutralizing their weapon.
Exactly.
So SC1 is essentially this active biological fire retardant. But they had to prove it, right? By making mice where the Rangers are at their posts, but their fire extinguishers are empty.
That is exactly what they engineered. The LDLR-KOSC1-MKO mice. The immune system was normal, the VRMs were there, but they snipped out the SC1 gene only within those specific macrophages.
And what happened?
Devastating structural consequences. The rupture rate jumped to 50% compared to just 14% in the control group.
Wait, 50%? Just from removing one protein from one cell type?
Yeah, it fundamentally destabilized the vessel. The smooth muscle cells lost their power, and there was a massive spike in inflammatory cytokines like TNF and IL-6. The wall just turned into a chaotic, inflamed mess.
And I remember reading they also deleted SC1 in lymphatic cells as a control and nothing happened, right?
Correct. The protective SC1 must originate from the macrophage park rangers to work.
Okay, we are about a third of the way through, so let's pause for a quick 5-minute mini recap for everyone tracking this at home. Bullet point 1, AAA is a lethal ballooning of the aorta.
Right. Bullet point 2, we have these live-1 positive VRMs, our park rangers, living in the vessel's outer wall to protect it.
And bullet point 3, they protect the aorta by constantly secreting a chemical fire retardant, a protein called SC1.
That's the chain of events so far. But here's a question for the audience. We know SC1 is the protective shield, but what exact biological disaster is it preventing from happening in the first place?
Yeah, what is actually destroying the vessel when the fire retardant is gone?
To answer that, we have to look at the microscopic plumbing. Bioinformatics pointed heavily toward a process called lymphangiogenesis.
Which is the creation of new lymphatic vessels, right? Angiogenesis is blood vessels, so lymphangiogenesis is lymphatic.
Exactly. In a healthy aorta, you have this controlled network of lymphatic capillaries that drain away metabolic waste and excess fluid.
Which is normally a good thing. You want fluid to drain.
You want controlled drainage. But without SC1, the genes driving lymphatic endothelial cells to multiply go into absolute overdrive. The researchers proved this using a DIR dye experiment.
Oh, this is the part where they injected fluorescent dye into the tissue to watch the drainage in real time?
Right. In healthy mice, the dye flows smoothly through tight tubes. But in the mice lacking SC1...
Let me guess. Total structural failure.
Complete failure. The dye aggressively leaked out of the lymphatic vessels directly into the surrounding tissue. And the volume of that leakage perfectly correlated with how large the aneurysm became.
So think of it like this. Imagine a city hit by a flood, and they hastily build an emergency plumbing system, but they build it too fast with cheap materials. The pipes multiply uncontrollably, they aren't sealed, and they are completely leaky.
That's a perfect analogy.
But they aren't just leaking harmless water. This leaky plumbing is actively creating a toxic microenvironment, right?
Yeah, what's fascinating here is that it isn't just a passive mechanical failure. The lymphatic endothelial cells themselves become dysfunctional, and when they do, they panic. They start acting as rogue recruiters.
So here is a question for you listening. If your emergency pipes are leaking fluid into your structural walls and panicking, what kind of unwanted guests might that attract?
Well, the panicking cells release specific distress flares, powerful chemokines like CXCL13.
CXCL13. And chemokines are basically chemical air raid sirens.
Exactly. And these sirens attract the heavy artillery, T cells and B cells.
The body's elite immune soldiers.
Right. Normally, they live in your lymph nodes to fight systemic infections. They absolutely should not be chronically active inside the delicate wall of your aorta.
Because if you bring heavy artillery into a fragile ballooning wall, the collateral damage is going to be immense.
Exactly. Swept up by the signals from the leaky pipes, the T and B cells swarm the tissue. And they don't just patrol and leave, they set up an unauthorized, permanent military camp directly inside the structural tissue.
Which is what immunologists call a tertiary lymphoid structure, or a TLS.
A TLS, yes.
Okay.
It's essentially a rogue fake lymph node right in the middle of an active aneurysm.
That is so wild.
And this rogue camp is hyperactive. It recruits those destructive circulating macrophages we mentioned at the start. They dump out enzymes, chew through the elastin, and the artery violently ruptures.
But in biology, observing a sequence isn't enough. How do we know for absolute certain that the leaky plumbing caused the rogue military camps to form?
They tested it by intercepting the cascade. They gave the mice a drug called MAZ51.
Okay, what does that do?
It's a VEGFR3 inhibitor. It chemically shuts down the wild proliferation of the lymphatic vessels. By blocking that, the leaky pipes never formed.
And without leaky pipes, no chemical sirens.
Right. And without the sirens, the T and B cell military camps never arrived. The tertiary lymphoid structures didn't form, and the aneurysm was vastly mitigated.
Okay, let's do our second 5-minute mini recap. Bullet 1, a lack of SC1 causes abnormal leaky lymphatic vessels to grow.
Bullet 2, these leaky vessels secrete signals that attract T and B cells.
And bullet 3, these immune cells form destructive camps called TLS in the artery wall, driving the aneurysm to rupture.
That's the domino effect. No SC1 leads to leaky pipes, which leads to rogue immune camps, which leads to the aneurysm.
So we have our crime scene and our sequence of events.
But we have a question for the audience. We are missing the murder weapon, essentially. How exactly does SC1 physically stop the pipes from getting leaky in the first place?
Right. How does one single protein hold back an entire biological cascade?
This is where they had to get down to the atomic level. They used mass spectrometry, which is a highly sensitive tool to map protein interactions. They used SC1 as molecular bait.
And what did they catch?
A very specific target. Fibroblast growth factor 2, or FGF2.
Okay, FGF2.
Yeah, the powerful biological engine that stimulates vessel growth. In mice missing the VRM rangers, freely circulating FGF2 was massively elevated.
So SC1 interacts directly with FGF2, but how does it neutralize it? I love this part. Here is the mental hook for you listening. Think of the SC1 protein as a massive, highly trained biological bouncer at a nightclub.
I like that.
SC1 has this highly specific region called a calcium binding domain. This domain acts exactly like the bouncer's arms. It reaches out, grabs the FGF2 molecule, and locks it into a molecular bear hug.
It's a molecular sponge. As long as SC1 is there, FGF2 is trapped. It can't overstimulate the lymphatic cells, but without the SC1 bouncer,
the FGF2 runs totally wild.
It runs wild, binds to the lymphatic cells, forces them to multiply too quickly, forms a leaky plumbing, and triggers the whole fatal cascade.
Which brings us back to our patient sitting in the exam room with a ticking time bomb and no options but surgery. How does this massive biological detective story actually help them?
Well, this is where we transition to application. The researchers looked at the SC1 bouncer, and they realized they didn't need the whole massive protein. They just needed the bouncer's arms.
The calcium binding domain.
Exactly. So they synthesized a tiny peptide, just 17 amino acids long copying that exact domain. They chemically modified it with an acetylation group to protect it from being chewed up in the bloodstream.
And they called it SPAT-17.
SPAT-17, yeah.
So they essentially engineered a microscopic bouncer in a bottle, and they injected it into mice with actively ballooning aneurysms.
And it worked perfectly. It had a 31-hour half-life, which is an eternity for a synthetic peptide. It naturally accumulated right at the site of the aneurysm, acting as a molecular sponge to trap a FGF2.
So the plumbing stayed tight, no sirens went off, and the TLS immune camps dissolved.
Right, the incidence of fatal rupture plummeted from 43% down to just 14%.
But wait, I have to play devil's advocate for a second. Don't we already have drugs that target blood vessel growth? Like cancer drugs. Why invent SPAT-17 when we have anti-angiogenesis drugs on the market?
The researchers actually tested that exact question against a broad-spectrum cancer drug called ponatinib. It was devastating. Ponatinib is a sledgehammer. It blocked receptors across the entire body, causing crippling hypertension, huge heart damage, and massive mortality in the mice.
Wow, so the systemic treatment was worse than the disease.
Exactly. SPAT-17, on the other hand, is a sniper rifle. It specifically targets the FGF2 pathway locally with zero cardiac side effects.
Ow.
It's an incredibly promising, non-invasive therapy.
Okay, let's bring this all together. Think about the sheer elegance of that for a second. Using the body's own defense blueprint to engineer a highly targeted drug. We tracked a disease from a disappearing park ranger cell to a missing chemical shield to wild, leaky plumbing and rogue immune camps, ending with a tailored peptide cure, SPAT-17.
It's a masterpiece of translational medicine and really leaves us with a broader perspective to mull over. Our bodies have incredibly specific, localized ways of maintaining peace in high-stress areas like the aorta. If a single protein from a rare tissue-resident macrophage can diffuse an aortic aneurysm by acting as a molecular sponge, how many other seemingly incurable diseases are just waiting for us to discover the local microscopic park rangers that already know how to fight them?
Right. Instead of using external chemical sledgehammers, the blueprints for the cures might have been written into our tissues since we were embryos. Such a wild thought. Well, thank you for joining us on this deep dive. Stay curious, keep questioning the mechanics of the hidden world inside you, and until next time, keep exploring.